Good morning, everybody. Welcome to day 2 of our workshop. Thanks for being here. We will start with our co-chairs going over what happened on day 1 and discussion of ideas for future collaborations. Just one quick announcement. If you could stick around at the end after we adjourn for a quick photo, that would be great. Otherwise, I will pass it on to our co-chairs.
Great. Hello. Should I go first?
Yeah.
Okay, great. So I guess next slide, please. You want to do the introductory comments or?
No, you send on to the next one.
We thought, so the main goal for this is actually just to kind of tee up the teaming that's going to happen and preparation for the next session happening right now. And so we thought a couple things could be useful. We made some synthesis of some of the different ideas, maybe not comprehensive, but we tried to put some pieces together that were in the presentations from yesterday. And we thought actually to keep this off, it'd be useful to kind of show some of the stuff from the notice of special interest that we referred to yesterday as a way in which some of these projects that we're going to be talking about can be funded. And one of the key things in not just reminding folks of that opportunity, but many of the words and concepts from the notice align very well with some of the things that we've been talking about here today. So that's really neat to show. And so we just pulled out some of the words from the sort of point of that notice of special interest, align with some of the types of connections we've been making. Next slide.
Yeah, maybe I can say a few words on this slide. So actually, this was quite interesting because in the NOSI, then the different institutes would bring in their mission statement or like their statement of interest. And it was actually really very interesting to review this again yesterday evening, where, if I may just briefly mention, this is from NIA. And a lot of the ideas we discussed yesterday were actually also apparently the interest statements of NIA, which is great. So, for example, they say, develop biosynthetic reporters of the hallmarks of aging. We talked about the hallmarks of aging, several of those. For laboratory animals, lab animal and human-derived organoid and cell culture systems, we talked about that. Develop tools and technologies to control and reprogram aging systems for accelerated or delaying aging. We talked about that a lot yesterday, and we have that also in our upcoming slides. Apply synthetic biology approaches for the development of biotechnology, biomedical technologies to track neuroscience-based interventions and preclinical studies. That came up a lot of times yesterday. And then also technology tools applicable to multiple morbidities in aging populations. Apply synthetic biology approaches for reprogramming and rewiring the aging and or disease systems. And actually that's interesting that they also discuss and say or disease systems, because in one of my breakout sessions that was a question, is it also applying to age-related diseases? So it seems it's comprised, and I think that makes really sense because they are very interconnected. But then maybe, yeah. So I thought that was really cool, that this really, we seem to be with our discussions very well aligned with NIA's thoughts, which is nice. And then Josh, maybe if you like to describe the next slide.
And I guess but the point of all this is really not that these necessarily anticipate all the things that we talked about yesterday, but just that the specifics that we've been talking about fit well into the big categories. That was the only thing I was going to mention about the NIBIB statement on it too. Next slide, please. Which is just that everything we've been talking about potentially, but I think that's not speaking on behalf of the NIBIB, but we can cross-reference later on if you have some details.
So if you have any further questions about the NOC and the mechanisms to apply to, you can reach out to me for EB, and I will put you in touch with my colleague at NIA for the NIA portion. Awesome.
Thank you. Next slide. Okay, you want to take this one?
Oh, yeah, so I mean, it's really an attempt that we did yesterday afternoon and this morning, Josh and I, to maybe summarize a little bit some of the things that we discussed throughout the day and in the breakout sessions. Maybe I stand on this side. One of the ideas that seem to have come up a lot is actually maybe finding amplifiers and reporters and or recorders for molecular signatures. And so these signatures can be many things. They can be biomarkers, they can be panels of biomarkers, or more specifically also phenotypes of specific cells. And these amplifiers may have a very high sensitivity because of the circuit systems that would be maybe implemented. And this could then help really kind of shedding light onto dynamic procedures during aging, temporal changes. It may discover many facets of heterogeneity and may or not discover also some noise. But part of that could also be the concept of a cellular sentinel. You know, having a sentinel for a cell when it starts aging, or describing the level of aging, and maybe at some point reaching a threshold where a sentinel would then trigger maybe you know, some either the amplifier, or that could also potentially trigger the therapy, you know, or like delivery. But we come to that in the next slide. But you know, we did discuss yesterday, we had this nice you know, kind of graphic here where we're looking at cell functions, its vitality, there's a maturation step, and then molecular readouts and biomarkers may actually reflect that the state of health where it still matures, peaks, and then may decline. So we actually really liked this graphic, and it came up in multiple different groups. So we're just showing your graphic here. And I think you know, hopefully we can use some of these SynBio approaches and implementations and integrations as readout also for a functional metric or performance, right? So not only looking at amplifying biomarkers or states, but also maybe bringing in kind of a functional readout that could be, say, a calcium release or some, you know, kind of metric that one could then kind of report this way. What will be interesting potentially is once we have integrated a SynBio element or circuit into a system, we might be able to perturb and then also see how the cells react to that, or the organoids. You know, maybe early on perturbations can be easily overcome with some kind of you know, healthy system. When does it come to the point where the aging cells cannot, you know, recover from certain perturbations? We also thought, and this was discussed a lot, what could be the systems we could try and integrate a SynBio system into, which is maybe one of the key and important questions to get started, right? And so we had a lot of discussions. You know, we would have to have, you know, model systems for aging. We talked a bit about maybe integrating at an early, say, iPSC state where it's still easy to do these performances. But Josh and I talked a bit this morning, and we also think it's possible for other more mature cells. These would be things we would work out. But actually, it is interesting. We could then look at aging or age-like processes. It could be senescence, but also where, you know, you look at aging and then maybe fibrosis, right? Because that's something that often happens as part of aging. And then we also thought that some of the models that Jennifer was showing yesterday, Jennifer Ellis's, were great, where you can also in a mouse model induce fibrosis. So we could actually look at some of these kind of phenomena with the SynBio systems, and potentially even changing, you know, what happens. It was also of interest to maybe think about organoids, iPSC-derived organoids, because again, those might be where we could integrate the systems earlier. But then one could, out of these iPSC that have the circuits integrated, one could make, I mean, neurons, astrocytes, microglia, all kinds of cell types may be interesting for the brain. But again, then also these more complex organoid structures where you then have multiple cell types in it. But that could also be another really kind of pointless discussion. What would be good model systems where we could try to get started with? These were just some ideas we had as kind of summary from yesterday, and we have one more slide that maybe Josh will kind of show about maybe some therapeutic angles, but I would like you to all think, or are there other things we would like to add to this list? So maybe we should go over the other slide first, and then we can come back to both of the slides and add things that you feel were important. These were just some starting points. Next slide, please. Sure.
Yeah, and these are just kind of like the synthesis of the things that we had. We got through all the materials. Thank you everyone for depositing materials onto the box folder. So the goal of these is really to tee up what we're going to use the rest of the hour for here. And that's just to kind of talk a little bit about these towards the goal of trying to pick at least 4 breakout topics that can be the subject of the discussion last time through. So we can certainly add more to it. I can talk from here, I guess. So this is like another big list of therapeutic concepts here, and all these are already posted to Box, including a fix to the typo on the previous slide. I don't know if you saw it, but I did. So the version on Box has these things if you want to be looking at them through right now also. And so just listing some of the ideas that came across in multiple discussions. Screening tools for drug development, drug delivery strategies, cell-based therapies of many flavors, including those for modulating the aging environment. Sensing and responding, delivery, homing, various modalities were mentioned. There were a few different mentions of things that had to do with like ways of shifting the state overall of the aging body. Synthetic parabiosis is my like super jargony thing, which is just like the idea that there are these like interventions we know work pretty complicated. Are there ways we can recapitulate those with an intervention that's simple and translational? Rejuvenation for targeted delivery, maybe to specific tissues, cell types, and chromatin engineering is maybe well placed up here too, as like another one of these areas where targeted action can be applied. The microbiome is another target that can be engineered conceivably. It can definitely be engineered, to affect aging conceivably. And then various different things having to do with looking at the mitochondrial axis and pillar of aging. Things that could be targeted to the mitochondria. Synthetic mitochondria were mentioned as maybe a both research tool and as a potential delivery modality, kind of along the lines of microbial therapies that are definitely translationally practiced now. Interrogating in general these kind of organelle scale contributions to aging, and again, that's like the connection between mitochondrial bioscience and maybe the synthetic biology tools that could enable that. Models and tools for interrogating and perturbing intracellular coordination and communication, one of the axes for applying these things. I think this is under therapeutic concepts, but most of these have a discovery through therapeutic possibility on these teams. And I think mentioned chromatin engineering and then synthetic biology for making diversifying natural products as maybe inputs into this drug discovery platform from the first couple of things there. Okay, so this is enough that you should be able to do all these things in like 3 years. I don't know. So, but I think this is the part we weren't sure how this would work. So I'll sit down. We should just all talk a little bit here and see kind of there's some natural gravity. I think our goal is not to make sure everything is comprehensively captured here because we have other ways of doing that. But if there's something you especially want to make sure is part of our discussion about what a breakout might include, please definitely mention those things, and then we can kind of try to organize ourselves.
Right. Yeah, maybe we can go one slide back first to the discovery. And I mean, are there other things we maybe didn't report that we thought would be very important?
I think it's in here implicitly in a couple of things, but it's not explicitly stated. And when I think of synthetic biology, I think of artificial circuits and that kind of direct manipulation of these things. And so there's some things like perturb or do this, and maybe that's a way of doing it, but I don't know if it needs to be called out more explicitly, but to me that suggests a research program because that it's at least concrete and would capitalize on the best of, not the best, but something that synthetic biology is doing and is kind of truly synthetic, and that aging biology, except in a few notable cases, just doesn't use those tools yet. And so that's what I've been focusing coming into this meeting. That's what I was thinking would be good. And coming out of this meeting, I'm still thinking that. So so I don't know. I think it's in there. You can use it for discovery, but I was thinking, just autonomous switchable systems that could systematically probe the underlying biology and or move it into different states and or those kinds of things. It's kind of what I was thinking about. And I think it's underlying a lot of this. It's just not, it's not just called out specifically.
So maybe, I just want to follow up with that, maybe you're thinking, this is what I think you're thinking. Maybe you're not thinking this, is almost have a cell go in. Because these signals, especially senescence, are so low, right? That is there a way that we could program a cell, ping a signal, and but then that cell then starts to like turns on a signal to start amplifying another signal. And I don't know if there's other ways we have multiple synthetically engineered cells in there, then that signal could actually be much bigger to report on something very small. Is that what you're?
Well, that's the amplifier idea.
But it's also
I
the
think,
circuits and the feedback and the.
yeah, no, I agree. I'm just thinking about just the fact that I still don't think we know everything that is happening. And can we create tools that are basically a cellular microscope to systematically just kind of blow through every gene in the genome, to turn it up, turn it down, do whatever, not be so dependent on external signals. I think I've told this story before, but the genetic system that was discovered that started really the genetics of aging revolution was the insulin signaling pathway in worms. And so it's this DAF2 gene, which is the insulin receptor. And then there's a whole kinase cascade, goes down, turns on DAF16. DAF16 is a transcription factor that gets phosphorylated, and then it migrates into the nucleus and turns on a whole bunch of genes, hundreds of genes. This has been known for more than a decade, the whole thing. And then somehow, we don't know what all those, we know what the targets are. We don't know which targets and which tissues actually make the worms live longer. To me, this is mystifying. And Ron, maybe you know more, and Max, on what the inside story. I don't know why we don't know that in excruciating detail, like exactly what's going on. So we kind of stop halfway through the process. You're going to design a drug. You don't want to design a drug to manipulate the insulin pathway. That changes everything. What you'd like to do is design drugs that are affecting the specific effectors that are making the worms live longer in a specific tissue. That eliminates all the pleiotropic off-target effects. We don't know those things. We don't know which ones we should be looking at. We don't know what reporters we would generate to find the drugs that are specific to those things. So I think that there's still, and that's just in worms, like what's going on in a human cell or any of these other things. So I think we're still a long way from the precision that's necessary to design therapies that won't screw up everything. And that's where I think synthetic biology at least holds some promise to surfacing those issues aggressively.
So I think part of the question there is how many cells do you have to be in, how many cells in the organism have to be engaged in aging, is their loss of function, to then have a physiologically manifest loss of function. And it might be different for a worm, fewer than thousands, versus us with trillions. So that's a problem. So, are there thresholds or tipping points to get manifestation? The other thing is, and I may have missed it, where's the model? Mathematical description of the circuit to engineer will have an outcome. So I think those are linked. First one, I don't know if any of you remember Stuart Kim. I used to be at Stanford. Kidney transcription factors. Very good project. So he and I had once discussed this question: how many cells do you have to engage? And if you made a chimera, short-lived animal of the same species, longer-lived, mixing what you have to have basically a circuit to the whole organism, multicellular. So, and it's just, yeah, I think maybe those are. I think it's the genes. Don't you need the equation to describe circuit? Yeah.
That's great. I'm thinking like this is kind of like getting to what we're hoping will happen after this too, is kind of getting to the detailed versions of some things. So this is only to make you notice holes that need to be filled in the subsequent conversations, I suppose. So that's great. And I guess what I'm listening to is whenever there's something we tend to talk about, that might be a good one for us to elevate for one of the breakout rooms here. So and I think it'll be tough to narrow any one of these down if within those breakouts too. So if you think there are two big themes within these two, that's another thing to mention here.
One thing we also mentioned this morning, for me, I think what's really great, we have a lot of really cool conceptual ideas. What remains slightly unclear for me too is maybe still how would we actually get started with something? Because I think if we had, for example, some kind of cells, but even with just that very first point where we have amplifiers, I mean, that would be huge, right? Where we could amplify an aging signature or something, right? Like, even though, but so, you know, but then, yeah, so I think, I don't know if there's also interest in sketching out some practical ideas or feasible ideas, or I don't know.
Yeah.
I mean, then Josh was saying that,
Maybe the next,
right.
I would think yes, but maybe the next,
Mhm.
again, the breakout. And I think, by the way,
Yes,
we can just move
Sure.
to that mode. We have like the 2 hours really for this and then that.
Right.
So the
Sure.
main challenge, I think, is like having an idea of where we should all go to have those conversations. That's kind of what our objective is at the moment.
Mhm.
Well, just building on that idea, a strategy would be, are there for first mover topics
Mhm.
Yeah.
that are broad?
Right,
Exactly.
And then that would create focus, like what is the first things that we would want to accomplish?
right,
Under epigenetics, under circuits, under the hallmarks or markers of aging or whatever. Because those will translate into the first grants that
Right.
or collaborations that will be meaningful.
right, exactly.
Because all these things are great.
Mhm.
Good. I have a question. So is there, so I managed to do some reading last night after a big. And I've read this, this paper that was published earlier this year. It was the ICE system. I'm pretty sure a lot of folks in the room have heard. So it's this induced double strand break system that was set up in mice to avoid damaging DNA while setting off the cascade of events in the cell that respond to double strand breaks. And they followed epigenetic changes and stuff. Anyway, so I read that paper and I learned a lot about, what, just it listed out hallmarks of aging and all of that and put it in the context of epigenetics. The one thing that stood out to me, having looked at epigenetic remodeling in the context of cancer, it seemed like aging looked like what happens when you give someone epigenetic therapy. Same signature genes come up. Oh, immune signaling genes come up. Oh, yeah, that's what we see when we treat, cancer cells with, HDAC inhibitors and polycomb inhibitors. And so I'm just kind of wondering, so this is just a question for folks with the expertise in the room. Is sort of understanding the similarities between, aging moving in either the forward or reverse direction, the parallels between that and cancer, is that a research priority that's relevant to, I don't know, something that could be included, or do we want to like not focus on that and focus more on, just sort of like wild type cells and just general aging?
Well, I think, certainly it's a really interesting connection to understand, and we don't fully understand. But cancer is also an age-related disease. Aging has with it a list of degenerative phenotypes, but also hyperproliferation as well. So I think understanding that would be the connection, right, between these age-related molecular changes and how that creates something permissive for
Mhm.
that for cancer was also very interesting and,
There's one thing I kind of noticed is that it seems that, so I also, going in the opposite direction, right? So epigenetic signatures that have been, found at higher frequency in cancer cells, higher H3K27 trimethyl, for instance. But it seems like one of the conclusions from this, so they actually reversed aging by, overexpressing the Yamanaka factors. But it seems like the shift away from aging towards a younger state included the gain of a lot of silencing marks. And you see the same pattern with cancer. So cancer cells do the same thing. They have higher levels of silencing marks
yes.
than the, non-cancer comparators. I'm pretty sure there are some other key differences that would distinguish a cancer cell from a de-aged cell, but I couldn't help but I'm really fascinated by those similarities, and
Yes.
that's why I kind of, so based on my quick reading in my 24 hours.
Right.
Yeah.
I think one thing that a broader way of thinking of the theory of aging that may or may not be true, and it's probably true for some tissues or whatever, is, and that where aging cells and cancer cells could share something in common, is they're both doing something they shouldn't be doing.
Right.
And so a theory of aging would be misexpression of genes in a cell that should not be expressing those genes, and that's what a cancer cell does.
Mhm.
So I think that could be a tie epigenetically, and that would go to this suppression, is maybe tamping down those bad things
Right.
again. So that's an idea. I would say that that's an untested idea. And we're just now starting to get the data. It's not my specific area, although we look at it work. If cells start doing with all the single cell work in aging mouse populations, that's just starting to go. A lot of tissue specific things, I think, be able to get that. Like if the kidney starts expressing skin related genes as things get older, then that would be consistent with that hypothesis. I don't know that those groups have reached any general conclusions, but it seems incredibly likely that that's going to be part of what.
Yeah.
Yeah, cancer cells and aging cells, they have many things in common. So the first, cancer hallmarks were proposed by Hanahan and Lambert 20 years ago. Then about years ago, people, in the aging field model use, cancer hallmarks, then propose, aging hallmarks. So many pathways, for example, DNA damage and repair pathway, some kind of immune system, many things are shared. So NIA, we are working with NCI, we have cancer aging committee. So we are very interested in the direction. In fact, we have a portfolio of cancer and aging biology, in our division. We are interested in that angle. So in fact, the ICE model you mentioned is probably a good model for synthetic biology, right? Because it induces some kind of small, cut in the DNA, then repair them.
Yeah.
That cut
Yeah.
induce epigenetic changes. But I think for epigenetic changes, it's hard because there are so many
Well,
modifications.
it's the way they did this, it was indirect, and I was very dissatisfied by the lack of explanation of, it's like DNA break, repair, epigenetic remodeling. It's like, well, there's a big gap there. There was no exploration of the specific, how those changes were triggered, but they measured it, they observed it. So I just thought that was interesting. I think, yeah, synthetic biologists could definitely, propose something a bit more controlled and well characterized, but it's a very cool paper.
I think it's no, yeah.
So maybe we should put those slides back on, do we? Or, Yeah.
Yeah, that'd be great. I was actually just looking at the hair.
oops,
At some point, what's gonna happen is someone who's not Yukit and I will be asked to who would like to nominate a topic to do to lead a breakout room or to be in a breakout room. So just be thinking if there's one you really want to elevate. People are raising some ideas here that I think fit to that, but that's what we're gonna try to do. If this is, like, what I kind of hear is there's more conversation we would have, we're not having it right now because we're waiting for the next thing, so.
Right. Yeah.
Any things that rise to the fore we didn't already mention, could be an organizational. We could click to the next.
Could we bring
Dwayne,
the slides
Dwayne,
back
can
up?
you-
I see Dwayne leaving.
Oh.
He's running our way.
Oh.
Wait,
That's funny.
try and find.
I guess, I think I wasn't sure, maybe it was already listed on the slides, but just thinking about what you had been saying earlier about understanding the cell types and, where these things need to be expressed and everything, but I guess in parallel, even assuming we had that information now, do we have the tools to then target, those cells? So having some kind of parallel research or track to actually, develop methods to do that would also, be useful,
Yeah, for sure.
Your slides are back up.
Thank you.
Thank you.
It's great.
Which, is this the right slide you want?
Yeah, we're kind of going back and forth between two, unfortunately, but for now, this is great.
Well, one room that I think should exist, but I
This
won't, contribute to would be this kind of artificial aging systems through human cell lines, organoids, microphysiological systems. I think that that's a potential breakthrough area.
I know, I think
And
that-
I don't know if we have the people who want to talk about that in this room or not, but if I was creating an umbrella initiative, that would be one of the thrusts that I would have. Can we, and we talked about what the limitations of that could be. Does an aging cell line have anything to do with aging? Can you create sensitized cell lines that are actually kind of pre-aged by some molecular characteristic? There's a lot of themes along those lines, so that
Right.
again, the bioengineering concept of moving to scale, screenability, all of those kinds of things are not going to happen in mice or humans per se, but they could happen in cell lines. So why not just go directly to human cell lines?
Yeah, I agree. I mean, a human cell line, and then one could have some aging, kind of accelerated aging, either induced by inducing senescence or mitochondrial dysfunction or some problems in autophagy, right? So one could induce some of these things into cell lines that have reporter systems in them, synthetic biology tools, right? So I do think that one could learn a lot from that,
Yeah, that would be a huge, just modeling aging in
Right,
addition anyway
right.
would be a huge
Yes,
benefit to the community. I think
right.
one really important part of this discussion will be, how do you quantify aging in this system,
Right.
essentially? So figuring out a way to quantify aging in that system would be, open up a lot of doors, probe aging now.
Is it-
Because capturing the first point, screening tools. So have you, maybe it could be model systems and screening tools, so have you
Mhm.
sell
Mhm.
model systems of aging
Uh-huh.
that then one could use eventually to do a more high screening
Yeah,
approach.
that's true. So model systems, right, could either be senescence induction, autophagy, kind of induction. So that, and we can think about other. But I mean, I agree, that would be nice having a model system that then one could use to screen drugs or interventions. At the same time, one can still learn a lot about the system, I guess.
Yeah, I really like that. That actually, I think, sort of kind of corresponds with this idea to advance, right, the field beyond where it is right now by doing like precision aging or de-aging. And then so I think your earlier point maybe spoke to like maybe the tissue level, right, the macro scale. But then I feel like this idea, developing cell lines and actually sort of poking around at very specific pathways is precision at like the single cell level. I really like that idea a lot. And then those, yeah, if you could establish cell lines where you are manipulating in a more specific way, right? So you can do irradiation, we can do all these things, but how about, epigenome editing with the goal of aging the cell, or, some signaling pathway, right? Throwing in a synthetic GPCR, right, to alter signaling that mimics the age state. So once you do that, I think not only could you screen for maybe anti-aging drugs, which, there's obvious interest there, but also drugs that have been suspected to advance aging, right? So sort of retesting some of these hypotheses that have been stated about the effects of chemotherapy
Oh, yeah.
and these other therapeutics, and sort of use this as a tool to determine, can we reproduce that effect in this model? And then, so what are some ways to mitigate that?
But that's actually interesting what you just say, because some of the things is, for example, we have talked about this in the past, where, so for example, like HIV treated patients, right? They often show early signs of aging and early age-related diseases, which has been a big problem because it's like a big population who are taking these medications. So, having a toolbox kind of like that could maybe test drugs for problems. Do they do drugs, which drugs that are used for other implications, which of those could potentially lead to accelerated aging, which would be bad for a patient down the line 10 years later, which is hard to look at in a human because it takes so long before these things manifest.
And then in tissues that aren't really assessed, like I know that there's a pretty, community is very convinced that there's a correlation between frontline breast cancer chemotherapy and heart. So when the patient does survive the chemotherapy, then their chances of suffering the heart are increased significantly.
Yeah.
And that could be, so it's like, well, what is the nature of that? It could be that you're like prematurely aging.
Right.
Right.
Yeah. That's important. Yeah. Like that.
But again, I think this would be really great, but we have to think about what would be the readout in a cell to tell you that, this cell is aged now. Because, we could look at senescence, if there's more senescence, but, that's one aspect of aging you could model in a dish. Or the only other thing I really know is that you could measure, the epigenetic aging in the cells in a dish or something, but otherwise it's a little complicated. I think this is something like in NIA with colleagues there, we've discussed a lot too. We wish we had something in a dish that you could use as a model to screen for things. It is complicated. Yeah.
Right. Well, I guess because we're still trying to develop global aging markers.
Yes. Yeah. So
Right,
maybe
so-
building the markers, developing those would be also nice. Yeah.
Yeah, but I think the promise of synthetic biology is to not say, oh, we have this cell line, maybe it will age, or we got this low organ, but to create them. So if the cells are not behaving in the appropriate way, modify them so that they are the right markers, even if that involves creating artificial systems that are circuits within that, whatever, or to use because I'm an evolutionary biologist at heart, to use selection to create a lot of variation and then select on the subset. And again, that gets to the scaling. But I just think that the scale of the problem is such that we have to scale it up. And I think we don't know what the tools are that are needed in order to achieve that. We need to build those tools. And that's a synthetic biology research program, as far as I understand it.
Yes. Yeah, I guess I like the way you said that too, because part of the challenge is with any model, like it's like we don't know what the right model is, right? That's like everything, right? Every application has that. I wonder if there are opportunities to like build more plausible models and then see if, okay, that's their models, but then across those models, what are the common drivers of like, what's the role of chemotherapy and like these other, maybe this is a, genetic cascade model of aging. And this one is something in GPCR perturbation model. We think they're plausible because they have some indicators, some markers in common, some aspects of senescence or that could be like more, I think this like plurality of models might have some power too above this, because they're all gonna be just facets, but then you can start looking at the connections across those models. Does that sound reasonable?
I mean, if you engineered the organoid that showed extreme mito stress, whatever that might mean. And then you try to figure out how to pull it out of that state with drugs or
Right.
genetic interventions or something like that. That would at least give you something that's a discovery platform. Then you could take that into a mouse.
Right.
Engineer a single thing, but if you could assay, 10,000 drugs or it upregulate every gene in the genome within that line, and then see these do this, these do that, then you start creating a system in this perturbed state that you then create a hypothesis that you could then test about aging. I just think the natural history of aging is going to run its course. Like, we can look at old things, we can ask these questions. At some point, we have to change we have to do experiments that are going to test what aging is and how to change it. And I think it's more than normal kind of historical genetics, like developmental genetics is a good model, because aging is definitely not going to be a adaptively regulated process per se. I do think that doesn't mean that there aren't core things that will be held in common that can be changed, but there aren't systems that are evolved to age specifically. And so it's a very hard system, whereas in development, you breaking it is sufficient for discovery. In aging, that's not going to be sufficient to discover the essential elements. You're going to have to fix it somehow. And I think that's a much harder problem.
So do you think like, in that case, would something like C. elegans be a good way to then look at this rather than, for screening purposes, rather than just a cell model, because it's a whole organism and you can look at different phenotypes.
I think, I mean, my own vision would be that both are relevant, right? Because you want the whole organism, but worms only are doing so much. To some degree, I think the translatability of worms is still going to be the cellular responses.
Yes.
Because they don't have hormones. They don't have they don't have immune systems. They don't have all these other things. So I think a parallel kind of approach. But I just don't think that I just think that this whole engineered human tissue thing is going to be this future state. The question is it going to be useful for aging?
Yes.
People are going to use it for a lot of other things. Studying inflammation. So my colleagues are studying it for inflammation response of artificial joints. Because that's what they do. They're bone people. And so and they're making organoids for that. That's cool. Bad knees are a big part of aging. But where are we going to be 20 years from now with those kinds of systems, knowing that we're going to start 3D printing, that we're going to be developing these things? How can we be pre-anticipating where that's going to be so that we're engineering the cell lines or creating those systems that will allow kind of the pace of discovery to start accelerating?
Good things. I'm sorry. Focus like an arrow on this, getting us into the rooms where we can
Right.
really dig into these ideas. So I hear two big ones that we've been talking about so far that are certainly, we've talked already about some. Special folks haven't talked yet. Are there things that we're missing from, because I feel like.
So I want to echo Patrick's previous point about the missing holes in the understanding of age, especially from a network perspective.
Yeah.
So even when we step back and look at the traditional model system, like yeast and C. elegans, fly, for studying aging, not talking about human cell lines, we still don't really understand what's going on there. Like, I mean, people already identified many genes that contribute to aging. However, how these genes interact with each other, what's the sequential orders, what's the causal relationship, can we write up an equation like Ron said to
Yeah.
describe this endogenous natural system even? So I think that's a big missing point, even in the most simple system for aging. So there, I think really the synthetic biology can help. There because I think it is sort of raised there, like to perturb the system to reveal the causal or correlation relationship between these network nodes, these important aging genes, how they interact with each other, what's the temporal pattern, what's the. So I think really the synthetic biology can help us do that because we can well control each of the single nodes and look at the other nodes, how they respond, how this will change. I mean, the lifespan, because we can really see the lifespan in this model organism. So I think that's like, I mean, this will help a lot of the future steps that we want to do with synthetic biology, because synthetic biology, we want to understand the system first, the endogenous system. But I think there is a big missing hole there, even like we don't even understand what's going on at the first place. So yeah, and so when we from the yeast to C. elegans, we get to this multicellular system, then the complexity suddenly increase. You have all these different tissues. Can we target a specific tissue, specific cell type, and then do this perturbation, right? And see how the network will change in different cell types in an aging animal. I think all this, like for example, some optogenetic tool to target specific tissues to drive their aging and see how this will affect the other organs. I think, yeah, even in the model system, even without developing a new organoid or new system, even the model system, I think the synthetic biology approaches will be very helpful. So this is more like an understanding of the endogenous network for aging. Yeah, so.
You know, I think a lot of the challenges people are articulating developing organoids and developing model systems for humans and getting synthetic circuits into those are things that people are working on broadly across specific, half a dozen labs working on organoids for the last decade. And they made progress, but I'd say that to spin those up as model systems for aging is, I think, kind of putting the cart before the horse there. We don't really have the synthetic tools completely ready to interrogate any models. That's not to say that the aging questions can't drive development. It's really guided tissue differentiation, using circuits for guided tissue differentiation, motivating those people right now to get involved. I've talked to Patrick quite a bit at this meeting, and I'm really kind of coming away with this, realizing that there's been a missed opportunity with synthetic biology in worms.
Yeah,
and I'm gonna plug that. I think that is a really great model system. I think I think a little bit fuzzy on what tools for expression are in C. elegans, but based on discussion, it seems like they're practical. I think the thing that we do really well right now is we think about ways to control expression in time and space. That's what the field has learned to do pretty well, I would say. And so, C. elegans offers this amazing system and control, presumably control things in time and space, and Readout on the tissue cell by cell basis the effects of these perturbations. So I would say my takeaway from this meeting is let's not sleep on C. elegans. And I think I'm not aware of too many research programs. Maybe some of you guys are, but it was. I think it's a great model system. It might be a good intermediate model system. We're getting to the right system.
okay, so we have 10 minutes.
Mhm.
And then I was gonna route now, see if anybody wants, I feel like we've heard about so far 3-ish ideas that seem to nucleate some discussion. Can read those. Maybe we can collectively talk about them.
Mhm.
But are there any other things we want to make sure we maybe haven't talked about yet, but they're close? I did want to focus it. The comment is, I think we've gravitated to a lot of SynBio enabled fundamental research as the real sweet spot, with long-term applications being also possible, but maybe there are some fundamental first steps for some of those things that are on the, what slide are we on? This one could also be fundamental on the second slide. I'm not sure if it makes it then indistinguishable from a fundamental project or discovery project.
One thing we talked about yesterday is also signaling, right? Using it's like some clue out of the circuit to then signal further, or using signaling to trigger responses. But again, maybe that's already too far along. Sure, but
Engineering engineering new circuits. Well, I guess it was already a little bit mentioned too. I think that's a really, I would think
Mhm.
this is something that came up yesterday too, that engineering in like a resilience pathway or something into cells, because with aging, you know, this ability to return to homeostasis is lost. So can you now engineer in some redundant signaling pathway or amplified resilience pathway, you know, to help to return to homeostasis, things like that would be interesting. That might be something to try and see elegance, actually, I think, because I feel like that in the field, isn't it true that you look at, you can already do these stress response, you know, look at stress response, and that C elegans that have better stress response are enriched for longevity,
right.
strains, right? So
Yeah, and if you think about, like, if you could invent a little machine that you could put in a cell, it would have many attributes of these things. It would kind of have a sentinel like function. It would be monitoring something that's in the cell that's going wrong, pretty static stress, something like that. And then it would send out a signal to a gene to upregulate, to help fix that,
Yes.
pre-anticipate or whatever. And then to Ron's broader point about a model, those kinds of feedback things can spin out of control, actually. So they're actually hard to keep regulated, right? In an equilibrium state. So is it even possible? I've been thinking about just kind of the one act, like, let's get a drive, have a circuit that can be flipped on and off and then turn something on. But the ideal would be to then have another part of the circuit listening to the cell and then utilize that same feedback. Then you'd have a self-contained anti-aging machine that you would put in there. That'd be super cool.
Very cool, yeah.
Talked about those as like maintainers of homeostasis.
Yeah.
That's one version, not the only thing you could do with that. So
Yeah.
it's sort of like internal state response of control.
Yeah, and then when you put it in a stressful environment, you'd be pushing that out to the machine. Equilibrium. That would be super cool.
Yeah, certainly. I mean, I still like the idea to parallel also continue to think about some human system in the end. I mean, that could be parallel, of course, but I would. So when you have the circuits now, what human cell lines do you have available where you have full circuits currently? I mean, is it that one could use some of that to convert that into a human aging system?
I think I'll guess you shouldn't worry too much about which cell types they've already been done in for the most part.
Right.
The benefit is oftentimes they work a little differently in different cells, but they more or less work in cells, which is, I think, part of the inspiration that they probably will work in worms with some doing also, right? They probably will work in other species with some effort. I know that's not true of planarian, because I had a conversation about how that is just not true. And I was like, but they're worms. We have C. elegans tools, and I got learned about how that
Not everything long and skinny is the same.
Yeah,
kind of
yeah.
work.
Right.
Anyway, but I hope that no one objected to Caleb's comment, which makes me excited about the possibility of C. elegans as being a fit for that. So anyway, I always say the same thing for all the human cells. It's not they're all equally easy or whatever, but circuits can be transferred.
Right.
Thoughts I had as we talked about that might arise from the C. elegans platform that could then inform the platform. Was what if we could engineer a synthetic physiological feature that disappears as it ages, introduce a gain of function that then becomes a loss of function over time. That's how we measure it, instead of trying to determine what an aged organ looks like. So we can follow in C. elegans or some other model system where we can observe aging physiologically. Crack this loss gain of function to loss of function phenotype that we engineered, and then we port it into an organoid model, or iPSC-derived model, and see what happens, to see if different triggers that synthetic triggers we can introduce cause that same loss of function. And therefore we can synthetically recapitulate aging without needing to see a biological or physiological response.
So from first principle, say this is the feature of the system, and synthetically create that thing, that module, and then put it in vivo into something, and then watch it age.
That would be very cool.
Maybe like biology for science for the biologists who would like to study adaptive.
Yeah, I think there's a lot that's missing in worms,
Yeah.
specifically. As I said, to me, it's your multicellular organisms, so you get those features, but they have determinate development and.
I used to be like stuff on loose lab and very jealous.
Yeah, no, there's the good things about it, but when you move to the mammalian systems, you pick up hormones, immune system, and circulation are the major features. That there's more, obviously, but those, when I think about the major things that drive the differences in those systems, those are the critical.
Would it be worth to have a breakout group or that's more focused on the in vivo systems rather than like the cell-based systems? I don't know if there's some people are applying symbiote more to like, in a mouse, you engineer something and test it, like the immune cell delivery
I
or
love
you like
it.
organoids?
I like
Okay. Well, maybe not then.
But it could, I mean, they're just mapping again from other fields, like there are certainly, cancer is not the only, but it's an instructive model system for how that can be done. And there's, everyone's probably familiar with the cell therapy idea where the cells go replace. You might be less familiar with, there's also a whole suite of symbiote tools that are more like a device that is the cells, and the cells can be not the same species even as the organism. And you can encapsulate them in a polymer network where diffusive contact can occur, but there's no cell-cell contact. So there's a number of teams that have shown you can do all kinds of interesting things with that approach. Mark Fussenegger is a name that you haven't been exposed to. He's done a lot of work with this kind of encapsulated engineered cells and shown you can use that to regulate satiety and hormones and diabetes and the other things that are kind of input-output, but at that system level between the implant, gets rid of the like, where did the cells go challenge. Don't know if that is relevant. That is cool.
I just have a quick question. Does anyone in the aging field work with zebrafish? Nobody?
You.
No, they live a long time. Yeah, there is a new fish model that Anne Renee has created at Stanford, that dies faster. And they've been working pretty aggressively and making a huge progress on that. The zebrafish live a pretty long time. Oregon says the zebrafish center, so I have many colleagues who work on zebrafish. Almost all zebrafish biologists study embryogenesis, early life stuff. And very few study adults.
Okay, I think we should maybe decide on how to split up. So I think we heard a lot of good stuff. Does anybody want to just say, I will be going to this, you don't have to be leading it, but just say, I'll be going to this topic, and then we'll kind of start and see if we have 4 or more or fewer
Mhm. Mhm.
there. There can be more than 4. We can like divide in the room. For example. Doesn't need to be just 4.
You ready for me to open the breakout room?
Yes, please.